My screen. Hey, there we go. Don't know why Zoom does that. It's always a mystery. Okay. So happy Monday, everybody. Today we're going to be starting the new second section. Just make sure we have. Oh, I'm sorry. Tiring weekend. Chat back up. All right. So essentially, what we did in the first section was going to give you a good kind of basis in Mendelian or transmission genetics, which is a very useful thing to have because really underpins a lot of how we think about genetics. But essentially what we're going to do in this section is break all of that, right? And so we're going to be talking broadly about three different things. One, sex determination and sex linkage, right? So that's what we're going to be covering today. When we talk looking at non-Mendelian patterns of inheritance, which, to be honest, at least for humans, for almost all traits, they're non-Mendelian. So all of that crap that you learn in Gen Bio 1 and high school bio and stuff about tongue rolling and attached earlobes and I don't know whether you can cross your eyes or not and stuff like that, it's all nonsense. It's what they call lies to tell small children is what I tend to think of it as. Hang on, let me just do something quickly. Give me just a moment. I've lost all my settings because Windows nicely updated me over the weekend and just kind of pretty much balked everything on my computer, which is super annoying as one can imagine. Oh, that's weird. So I'm just trying to look at see how. Oh, well, that's fine. Oh, is that it? No, that's not it. Oh well. Doesn't matter. There we go. That's the tip. Cool. Sorry about that. I was just trying to figure out how to do something which I'd always had done. Anyway, so by and large, I think it's pretty fair to say, other than things like single gene genetic diseases like cystic fibrosis or sickle cell disease, stuff like that, most traits are non-Mendelian in one way or another. And then the kind of most non-Mendelian thing, and this is arguably one of the hardest topics in transmission genetics for students to get, because there's a lot of math involved, is linkage. Not right now, Isaiah. I'm just chuntering on camera. I'll be on the PowerPoint in a little bit. It'll be PowerPoint 7, I do believe. Be there in just a tick. And what else? Oh yeah, linkage. And that essentially breaks the law of independent assortment, which we learned about in the last section. Okay, now I'll go on to the. Ooh. All right, let's not worry about that just for now. All right, so I always like this picture because it kind of gives me a bit of a segue into a history lesson, right? Because I like history. History's fun. And this, actually, does anybody know who this is? There you go. Question for the class before I tell you.
Is that the Romanov family?
It most certainly is. Oh, it's in the notes underneath. But yes, you're right. That
it doesn't say Romanov.
No, that's right. Good job. You're right. That's the Romanovs. So this guy here with the very impressive mustache is Tsar Nicholas, and oops, move that around. His wife, obviously, and these five are their children, and so they were in power. Tsar being kind of the Russian king, essentially, when the Bolsheviks overthrew the Russian royal family, and they went into hiding. There was a bit of a civil war for a while between Red Russians and White Russians, and these being White Russians, but I don't know why they're called that. But anyway, this chap down here, Lexi, he was a hemophiliac, right? Unfortunately, I guess, kind of depends how you think about it. He didn't live long enough to have issues with hemophilia, because all of them were executed by the Bolsheviks when they caught them, I think in Siberia. And actually, there's a big, I don't know what we'd call it conspiracy theory, because it's not quite a conspiracy theory, but until recently, their bodies weren't found. And so there was a lot, I think it was Catherine, I can't remember, one of the daughters. They always thought, a lot of people thought that she actually had survived. And so there was like the heir to the Russian throne was still around, or at least her descendants were. And then they found them all in a shallow grave in the forest somewhere in Siberia. So that put the end to that. Anyway, we'll be getting back to this later. Have a look at European royal family trees, which is a bit disturbing, but fun to look at nonetheless.
Oh, so I think Alexander did suffer a little bit, but Rasputin took him off a medicine that made him bleed more and made him less stressed out, which helped his hemophilia.
Oh, okay, interesting. That was Rasputin, did you say?
Yeah, Rasputin. That's why he became known as like the mystic healer, because he actually apparently did somewhat cure his Alexia of hemophilia.
That's really cool. Thanks, I didn't know that. I know Rasputin was a somewhat dodgy fella who got involved in all kinds of things.
He was well known with the court ladies, if we say it that way.
Right. Through his advice and good sense of humor, I imagine.
Yeah, we'll call it that.
All right. Anyway, moving on. So this is a great example. We'll come back to this later. This is really a good example of sex linkage or sex-linked inheritance. First, we're going to go through sex determination, which is kind of basically an oddity that depends on sex chromosomes or differences between sex chromosomes. And so, yeah, that's just a neat picture. Not really sure why that's in there. Essentially, the word we use is dioecious, right? Dioecious equals sexually reproducing. There are other forms of reproduction typically used in like insects and invertebrates and stuff like that. If we say dioecious, it means they're reproducing sexually, males and females. Or even, I mean, at least in my lab organism, C. elegans, hermaphrodites and males, which is kind of weird. But worms are kind of weird too. So when we're talking about sex determination, there's several different ways in which this can be determined. And this is not, some of these overlap, right? So there's a lot of interaction here between different components depending on the organism. And so one that you've probably all heard of it's alligators. I guess that's a Mississippi alligator by the name, where incubation temperature of the eggs determines the sex, right? So the female can actually manipulate the sex ratio of her offspring by varying the amount of kind of covering over the eggs, because they bury them in the sand and kind of like pile stuff over them to maintain temperature. And so depending on that temperature, and you can see here the sex ratio, so 50-50 for whatever kind of thing that is, turtle, tortoise, maybe. I'm not much of a taxonomist.
Alligator snapping turtle.
Oh, that's snapping turtle?
Yeah.
Oh, cool. Those things are vicious.
Yeah, they can be.
Yeah. I don't know where it was. Maybe it was San Antonio Zoo. I can't remember. Maybe it was at Atlanta Zoo Aquarium. They have one, and it has like this tongue, which it waggles around, and it looks like I don't know, like a fish or something. So a bird will come in, and then it will just go hunk. At least, obviously not in the zoo. Even as babies, I mean, I can imagine. Like actually, little alligators are mean. They just have tiny sharp teeth rather than really big sharp teeth. Anyway, so actually alligators are kind of even more of a weird one. Essentially around 33 degrees Celsius, which is obviously fairly warm, they have 100% males produced, right? And it's a very steep decline either side of that. So just a degree or two makes a huge difference, which is kind of weird, really, if you think about it. I don't actually know the mechanism for that. My guess would be some degree of temperature sensitive control of gene expression, right? Proteins which are functional or non-functional at different temperatures, which is pretty cool. So when we talk about sex determination, one of the things that we need to talk about are autosomes versus sex chromosomes. And so autosomes are all of the chromosomes, and we have 22 or 44 essentially, if we think about homologous pairs, which have nothing to do, well, yeah. It's hard to say that they have no role in sex determination. In humans, yes. In Drosophila, not so much. But sex chromosomes is a lot easier to define, right? So essentially they're different, either in size, number, or quite often actually size goes hand in hand with gene count as well, right? So in humans, the Y chromosome is, I'm kind of sad to say as a guy, rather stunted, right? There aren't very many genes on the Y chromosome in males. And there are many funky ways in which animals do this, right? So plants, I guess, as well. I don't know much about plants, to be honest. So typically you have homogametic versus heterogametic sexes, right? Homogametic, where you have two of the same sex chromosome. Heterogametic, where you have two different or maybe even only one sex chromosome, right? So a good example would be this one in the middle, which is XXXO. And the O stands for nada, nothing, absolutely zero, right? And so these, and one of the, it's not C. elegans, that's different, but one of the parasitic nematodes that I worked on actually used this as a means of sex determination. Essentially, the second X chromosome was destroyed in sperm. So any sperm that, pardon me, inherited an X chromosome as a result of meiosis, that X was destroyed. So there's just nothing there for the sex chromosome. Sperm in those species don't have X chromosomes, only the oocytes do. And then you have XXXY, and birds are kind of weird in many different ways. And they also have this kind of heterogametic kind of deal, but it's the other way around. And so in mammals and some insects, the homogametic is female. In chickens, it's the other way around. So in chickens, the heterogametic sex is female, which is kind of weird. And so essentially what that means is that you'll always end up with, if nothing else changes that, right? So no environmental deal, no sex preference, right? So if you look at, for example, in China and India and some other countries, where in India because of inheritance rules and in China because of the one child rule, males were favored. And there's lots of different ways in which you can favor that, either by selective abortion or just neglect. And so there's essentially an unbalanced sex ratio in those countries. There are more men than there are women, which has all kinds of weird effects on society. It really does. One of the cool things too, though, is that even though humans and Drosophila, for example, have the same XXXY sexes, right? XXs being females, XYs being males. The mechanism by which those determine sex is completely different, right? So essentially, they've both ended up at the same kind of outcome, I guess one could put it, but have got there via very, very different routes. And so this is a great example of convergent evolution, right? Where you start at different points and through different means end up at the same outcome, essentially. Okay, so I'm going to, oops, that was the wrong thing. Can you hear me okay? There we go. I unplugged the wrong thing. Okay, so this is actually a fairly advanced kind of topic. So I'll go through it steadily. You know, I won't give you a chance to ask questions. And one of the reasons why I like this and spend a bunch of time on it is because it's a very elegant, and as you probably know already, I like elegance when it comes to genetics. Elegant way of showing how you can achieve an outcome at a molecular level, right? So we're talking about a very kind of gross physiological outcome, right? Either male or female flies. And you've seen yourselves in lab, they're really quite different, right? They are different in size, they have different pigmentation patterns, they have different stuff, like the whole sex combs deal. So obviously different reproductive tracts as well. So that's underpinned by a very neat mechanism. And so essentially, let's Drosophila. And one of the reasons why I want to spend time on both this and essentially this is that then it's a really cool thing to contrast to how as humans we determine sex. And again, just talking about biological sex here, not going to get into the much more complex subject of gender and other things like that, right? So it's a bit of a simplification, but it's a manageable one, essentially. So in Drosophila, they determine sex by the ratio of sex chromosomes to autosomes, right? Right. The Y chromosome in Drosophila does very little. It is involved in sex-linked traits, such as sperm production is one that comes to mind, but it isn't involved in determining the sex of the fly. Right? That's a really important point to make because it's very different in mammals, right? So really what flies are doing, and this happens at every single cell, right? So this is a very micro level, well, not micro as a microbiology, micro as in small, mechanism. Whereas in mammals, it's very much a whole organism, large scale mechanism, right? So we've got that difference too, which is really cool. And so essentially what we have, just get this up here just to make sure I don't get anything wrong, is we have an 1-to-1 ratio is female, and this is X to A, where autosomes are A and sex chromosomes that matter in sex determination are X. And in males, we have a 1-to-2 ratio. Right, just give everybody a chance to kind of copy that up. So I grab my coffee. All right, all good so far? And so one of the oddities with Drosophila is that you can actually have, they can have really weird, like ploidy levels. It's a very strange thing, and flies are kind of weird in many ways. But as long as the ratio stays the same, the sex of that cell is the same. So you could have potentially, right, I don't think this actually happens, but potentially you could have 10 X chromosomes to 10 autosomes, and it would still be female. Or 5 to 10 or 10 to 20, and it would be male, right? It's the ratio that matters. And essentially what we have in lethal. We have one key protein, which is called sex lethal. Or SXL. Right, obviously, if that reason why it's called that is if you don't have a functioning copy at all, it's a lethal mutation. So we have one key gene. Actually, let me get this off like so. There we go. Ah, crap, I can't see what I'm doing now. Make sure I point in the right way. There we go. Don't worry about pointing up at the ceiling or anything like that. Okay. So if sex lethal is on, basically have pro female characters or characteristics, and this suppresses, that's what this kind of funky T-shaped thing, this is a genetic sign for repress or suppress, suppresses male traits, e.g. dosage compensation. And dosage compensation is essentially dealing with the fact there's a different number of X chromosomes in males and females. So in flies, Well, because they only have one X chromosome or half the X chromosomes, essentially. Expression from the X chromosome is boosted, right? In humans, all you ladies in the audience, the opposite occurs, right? So in every single one of your cells, and in cats as well, female cats at least, one of your X chromosomes is silenced, right? So even though you have two X chromosomes, functionally you only have one, which is kind of neat. Which one gets silenced is random as well, which is even cooler. So lots of cool things about being a lady. Anyway, so that's what sex lethal on does. And when it's off, we get male stuff promoted and we don't get any activation of the female traits or the kind of processes that lead to female characteristics. Right, so this is the general kind of view, right? This protein on, right? And on in this case means expressed, right? Could mean other things in other contexts, but on in this case means that this gene is expressed and this protein is produced. If it's produced, it promotes expression patterns that lead to female stuff, like reproductive organs and so on and so forth, growth pathways, and suppresses the male characteristics, particularly of dosage compensation. If this doesn't get expressed, essentially that allows these male characteristics to develop without any kind of inhibition, right? There's nothing around. And similarly, it isn't present to turn on the female pathways. Does that make sense? Because this is important. I need you to get this before we talk about how this works. Yes, that's right. So the ratio of X to A determines whether sex lethal is on or off. I just wanted to put this up first so you kind of get the bigger picture before we get into how this is controlled. Yep. You got it, Kaylin? I'll be posting this up too on Blackboard as well. Awesome. So the question is, okay, how does this ratio translate into this? How does this ratio translate into lack of this, right? And the key is two things that are produced one on the X chromosomes, one on the autosomes, right? And so on the X chromosomes, you get, and this is, I really, how I can put it, it's like giant pandas with me. Drosophila people call their genes silly things, right? And it's just annoying. See? Well, actually, these are like pretty tame. There's actually a gene called Sonic the Hedgehog. And there's another one which I saw at a conference, which some guy named Shaven Baby. Anyway, so, oh, I don't know. I think it's funny, I guess. I just find it annoying. It just gets ridiculous. Now in C. elegans world, we're very regimented. We have 3 letters to play with, maybe 4 now. Yeah, I guess so. And so it's actually, I've had lots of fun arguments with Drosophila people about like which way is better. But anyway, that doesn't really matter. And we have, this is Deadpan, not Deadpool. My kids want to watch Deadpool. I'm really not entirely sure about that. It's funny, but it's very, very rude as well. Anyway, so essentially what's happening is the balance between these two. So, and this, I'm sorry, I have to apologize ahead of time. I'm someone who works on genetic pathways. So for me, this is like, yay, you get to talk about genetic pathways. Woo! For a lot of you, like, what the hell is he talking about, right? Which is understandable. So essentially, what we have is a quantity deal. So Systalis inhibits deadpan. Deadpan's role is to inhibit sex lethal. So in females, we have lots of Systalis, or essentially an equal amount of Systalis, because we have a ratio of 1 to 1. We have the same amount of this as we have of this, right? So Systalis is able to inhibit deadpan. And so deadpan does not, let's move that a little closer. There we go. Deadpan does not inhibit sex lethal. So sex lethal is on. There's also a little bit of a kind of auto-regulatory deal going on there, but we won't worry about that. So we have an equal amount of Systalis and an equal amount of the inhibitor of sex lethal, which is called deadpan. These two essentially then cancel each other out, which leaves essentially this. Right? So sex lethal is something like largely unmolested, gets expressed, does its stuff, equals female. That's in females. Now in males, what do we have now? We have an unequal amount of Systalis to deadpan. Crab sticks, I didn't leave myself enough space. Let's move this over here. So we have essentially, right, a small amount of Systalis. My laptop's like that far away from the whiteboard now. And the same amount of, I keep wanting to call that Deadpool for some reason. I don't know why. Right. So there's not enough Systalis to inhibit deadpan. So deadpan inhibits sex lethal. And that's how you get male flies. So really what we're looking at is, this is like the switch, right? Sex lethal is a switch. If you flick it on, you get females. If it's not turned on, you get males. The thing that controls that switch is the ratio between these two, they're kind of like competitors, I guess one could think of it as, right? If we have an equal balance of these two, sex lethal stays on, right? It's not turned off. If we have an imbalance in these two, because we have an unequal amount of X chromosomes to autosomes, deadpan kind of gets the upper hand, one could think of it as, and does its job of inhibiting sex lethal, turns it off. When the switch is off, you get males instead. Questions? It's a bit of a kind of advanced topic, to be honest. So don't worry if you don't get it like first off. Yeah, the ratios are absolutely the key, right? So you can actually get kind of weird and funky like intermediates in flies if you get something that isn't a 1-to-1 or a 1-to-2 ratio. But as long as you have equal number of chromosomes, autosomes to sex chromosomes, you'll always have females. If you have a 1-to-2 ratio, sex to autosome, you'll have males. And it's all down to a gene that's on the X chromosome called Systalis, and a gene on an autosome, I actually don't know which one, but one of them, called deadpan. So the ratio is kind of detected, right, in a sense, by the relative amounts of these two proteins. If you have more deadpan, deadpan isn't inhibited, and so it is able to inhibit sex lethal. I'll take a picture of that while you cogitate. My camera. That's actually a really good question, Stephanie. So no, it's actually a very precise deal. So if you go outside of those two ratios, you get either intersex flies, right? So if you're between 1:1 and 1:2, you have intersex flies. If you have outside of those two ratios, you have, I don't even know what they're called. Like, I think they're called like megasex flies or something like that. They're like super males or super females. And so they're different. It actually has to be very precisely 1:1 or 1:2 to get the males and females. Let's see if I can so quickly. Doop, doop, doop. Oh, meta females. There you go. Yeah, super, super weird. Where are we at? Let me just share my screen. Yeah, there you go. So as long as you have a 1:1 ratio, you get normal females. As long as you have 1:2, you get normal males. Between that, it's intersex. Outside that, it's meta females. What meta females are, I don't know. I actually haven't got further than that. They're not mega females, they're meta females. Oh, so in Isaiah, that's actually more a human sex ratio issue. And it's, I don't think it's XYY, it's XXY, which is Klinefelter, right? And so that produces a very strange kind of mix of traits, which we'll get into in a tick. Okay. So that's a little complex, but it's a really cool. No. Don't worry about that. Again, that's just kind of for added information. Really, what you need to know is what is in the PowerPoint and what we cover in class. I'll get my coffee. I just put that up there because it's kind of weird, right? That you can have these intermediates and kind of things outside of that. Now, in humans, it's very different, right? And essentially, and this is a cool thing, I've actually done a fair amount of reading on this because it's just kind of weird, right? And some of you, I think, already know this. Up till 6 weeks old, you are neither male nor female. So up to 6 weeks, you have both. Post-fertilization, I guess. Fertilization, yeah, got that right. Both proto-female and proto-male reproductive ducts. And those are Müllerian ducts for females. Well, proto-female tissues, and Wolffian ducts, named after people that discovered them, essentially, for proto-male tissues. Right, so essentially it's undecided, right, at that point. And you have, so these are kind of regions of the developing embryo, which will later on in development through many kind of changes in gene expression, like will turn into, in females, will be the upper part, the vagina, the uterus, and the fallopian tubes and the ovaries, right? So kind of that part upwards, essentially. And in males will turn into the testes, epididymis. Not sure about the penis or not. I actually don't know. That's kind of like a urogenital deal as well. It's a little bit weird. But anyway, they are essentially have the potential this way or this way. And so at 6 weeks, there's a change in transcription patterns between males and females, which determines which way you're going to go. And that comes from the Y chromosome. So remember in Drosophila, yeah, that's right. And the clitoris and the penis are also largely kind of derived from the same stuff. It's just different degrees of growth, essentially. It's really kind of urogenital development is super interesting. I don't actually cover that much, at least the genital development in my dev bio class, but I do go into urinary development because that's also super interesting. Anyway, so essentially what happens is a gene on the Y chromosome gets turned on. This is called the SRY gene, or sex determining region, right? And what this does, and oh yeah, I actually forgot. In Drosophila, the Y chromosome is not evolved at all. So if you're male, you'll be heterogametic. So you'll have one copy of SRY. No, actually, female mammals, homogametic, no copies of SRY, lowercase because it's a gene. So if you have two X chromosomes, you don't have a copy of this gene. It is only present on the Y chromosome. And what the SRY protein does is it turns on a whole developmental program. So this is essentially a protein, and we'll talk about transcription factors later on in the course, but it recognizes the genes which are all involved in male development, particularly testes development, and it turns them on. And so at 6 weeks, SRY is expressed or turned on. And the protein SRY itself turns on genes involved in testes development. And testes secrete two important sex determining hormones, right? One is testosterone, right, which you already know. And testosterone is often called a sex hormone, but it's also actually kind of, it's a hormone that does a lot of different things, right? And sex determination is one of them, right? But it's not its sole role. It actually does a lot of really, really interesting stuff. One is testosterone. And that is responsible for many male traits. not just primary, but also secondary sexual characteristics like bone density and muscle mass, things like that, facial hair, or hairy backs and baldness as well. It's actually tied to testosterone. Don't know what that says about me, but anyway. And also something called the Müllerian inhibiting or MIF, Müllerian inhibiting factor. It's got lots of different names. And what this does, remember at the beginning I said there's two sets of ducks, Müllerian and the Wolffian. Müllerian are proto-female, Wolffian are proto-male, and they actually lie just behind each other, right? Kind of going down into what will become the urinary tract, essentially, right? So they just lie next to each other, and you can only have one. It's like Highlander, right? And so what MIF or MIS does is it promotes the degeneration of the Müllerian ducts equal male, essentially. Whereas in females, SRI is not present. You don't have that gene, right? It's one of the few differences genetically or in terms of genes between males and females. So that whole program never gets turned on. You have all those other genes, right? So as females, you have the testosterone, well, the genes involved in producing testosterone from other sterols and stuff like that. You have a lot of the other genes involved in male development, but they just don't get turned on, right? And so if they don't get turned on, right, the testes don't secrete the Müllerian inhibiting substance. So the Müllerian ducts develop, and if those develop, essentially the Wolffian ducts die from neglect, right? So they don't have anything promoting them, so they just kind of peter out on their own. And then by 8 weeks, as an embryo, the change has been made. So it's a very quick program. It happens very fast, even though there's nothing whatsoever recognizable as testicles, right, on that embryo, right? It's still like a little, kind of bean, like about that big, give or take. It's very small, or maybe a bit bigger. I'm not much of an anatomist, but oh, so sex lethal, that's totally different. That is Drosophila, right? So Drosophila count the ratio between sex chromosomes or X chromosomes and autosomes. In humans and other mammals, it's all about whether or not you have a Y chromosome. If you have a Y chromosome, you end up with testes and being a male. If you don't have a Y chromosome, you don't get testes, and so you end up being a female. Right? So they're two very, very different mechanisms. Oh yes, very much so. In fact, not so much species, but definitely every different genus has its own kind of wrinkle or different way of doing this. So it's really weird. It's also super funky how these things evolve as well. But I want to spend a lot of time on these two because they are excellent examples of how you can end up with the same outcome essentially, even though obviously a female fly is different from a female human, but you get there via very, very different mechanisms. And really at the end of the day, it all comes down to what genes are expressed and what genes are not expressed, right? And so really, when we get into the second half of the course and look at kind of the nitty-gritty of molecular genetics, that's essentially kind of where we'll be heading. All right, and I've chunted on enough for today. So yeah, we're a little bit, took a little longer than expected, but it was worth it. So we'll finish this off on Wednesday and then get into non-Mendelian inheritance patterns. All right. Have a lovely day, everybody. Study hard as always, and I'll see you on Wednesday or Tuesday, depending on if you're in lab. Catch you later, everybody.
Have a good day, Professor.
You too.